All right, let's get started. Thank you for joining us today for another round of our webinar series on investigations of biomedical issues and how they relate to human disease. Today, it will be about ion channels. First, I want to thank Gary Watkins, Director of Marketing here at AnaBios, for organizing and put together this webinar series. And as a quick intro for those who have not joined us in the past, I'd like to introduce AnaBios. AnaBios is a unique contract research organization and biotech company based in San Diego. We recover human organs from ethically consented donors and use the tissue or cells to conduct physiological assays in which we can test preclinical compounds for drug discovery projects. We've essentially redefined first in human. And working with AnaBios is as close as you can get to a clinical trial without actually doing a clinical trial. For the electrophysiologists in the audience, we regularly patch clamp human DRG neurons, human spinal cord slices, and human cardiomyocytes. In the download section of the webinar panel, we have uploaded the human DRG brochure, which you are welcome to download. Also note that there's a question panel in which you can post questions, which I will read out loud during the Q&A session at the end of the talk. Okay, next slide. And for those who are interested in cardiac electrophysiology, we'll be hosting a webinar on July 1st, presented by Dr. Nadia Abi-Georgis, who's AnaBios' VP of R&D. It will be a presentation of the data recently published in Nature Scientific Reports on the role of the late sodium current in cardiac arrhythmogenesis in human heart. This will be the last webinar of the summer because we'll be taking a hiatus in August and restarting the series in the fall. So without much further ado, it's my great pleasure to introduce today's speaker. Dr. John Del Rosario originally hails from the Dominican Republic, and I believe Dr. Rosario will talk some about his journey, which can be informative for the younger audience members getting started in biomedical research. Dr. Rosario received his master's from City College of New York and did his PhD at Rutgers University Medical School. He did his PhD research in the lab of Dr. Tibor Rojas, where he studied the GPCR-mediated regulation of Piezo2 and the structural characterization of inhibitors of TRPV5 ion channels. Following that, he has joined Washington University St. Louis as a postdoctoral research in the lab of Dr. Robert Gereau, who has previously presented an AnaBios webinar on his pain work. At WashU, he is studying the cellular and molecular mechanisms that contribute to the development of adaptive and maladaptive pain plasticity. Dr. Rosario has been supported by multiple NIH grants throughout his career. Today, Dr. Rosario will present on his TRPV5 and Piezo2 work. The presentation is entitled Piezo2 and TRPV5 Channels, a study of the endogenous and exogenous mechanisms that modulate their activity. Please take it away.
Thank you so much, Research, for the kind presentation. And also, I want to thank the organizers of AnaBios for giving me this opportunity, this platform to be able to show my work, and everybody who is attending to this presentation. I'm really happy and honored to have this. Thank you, Grace, as well. The title of my presentation, Richard already spoke about it, and is going to be discussing about Piezo2 and TRPV5 channels, something that I did in my PhD work. But before moving forward with that, I would love to just give a little bit of introduction of my educational journey. So I originally was born and raised in the Dominican Republic, and then right after high school, as you can see here, I immigrated to the United States, and I attended the Hostos Community College, where I took college classes and ESL classes because I didn't know how to speak the language, so I had to get in there. Right after that, then I moved to City College of New York, in where I did my bachelor's in science and biology. From there, I went to University of Maastricht in Netherlands to do an internship in molecular neuroscience, studying pain and also a lot of molecular techniques there. Then I went back to City College when I studied neurodegeneration. I did my master's there in C. elegans with Dr. Isaac Mano. Then from there, I moved to Rutgers University when I did my PhD in biomedical sciences in the lab of Tibor Rojas, when I work ion channels, neuroscience, and signaling pathways. And now I'm currently a postdoctoral fellow studying ion channel neuroplasticity and pain in Dr. Gereau. And without further ado, we're just gonna move into today's talk. So today's talk is just a little bit of Piezo2 and a little bit of TRPV5 channels, and all this data has been published. So if you really want to learn something that I don't speak here, you can definitely go to the papers and we'll be fair and get more information about it. So we're going to start with Piezo channels and how they are regulated by couple receptors, and then we're going to move the second part describing how exogenous nova inhibitors of TRPV5 channels. So first, I'm going to start with introduction. So Piezo channels are mechanically activated ion channels expressed in many tissues. It was firstly described by Dr. Arden Pardaputian in a 2010 paper he published in Science in describing the identity of these channels. They were expressed in many different tissues, as you can see here. Later on, Dr. Bailan Chen from China, he was able to describe the first structure of Piezo1 and Piezo2. But as you can see here, these are very large channels. They about 38 transmembrane domain. They have they form a trimer, and it looks like a propeller-like structure, as you can see here. These channels has been extensively studied in the sense of biophysics, how they are regulated by and how the C-terminal can regulate the activity, the beam domain, because how they are embedded into the membrane. And there is still a lot to know about that. We're still looking into understanding a little bit more of how they're opening and gating. Although we know that they are intrinsically mechanosensor, there is still more information to assess about what is their important mechanisms of activations for these channels as well. What we really know is that we express these channels in hex cells, like for example, or you can also express it in neuroblastoma cell line, and you give stimulus, specifically mechanical stimulus, as you can see here. We can definitely get a mechanical probe, and you can elicit a mechanical response, which is displayed here as these inward deflections right here, and these are mechanically activated currents. The stronger the stimulus, the bigger will be the current that is coming through the channel, and you can quantify it here. So this is the way that I actually all my experiments, and the way that I will be describing all these experiments as well here in the presentation. So Piezo2 channels has been described to be important for many physiological functions. For example, it's important for lung expansion, for skeletal morphogenesis. It has been shown to be important for mechanical age, baroresection, and also recently has been shown to be important for mechanical pain in the sense of allodynia. Also, it's really important to show that it's important for proprioception and as well from light touch. That's all has been shown not only in humans but also in mice as well. So I want to point out an important thing that that humans who are born with mutations in these channels in which this protein is truncated, we can definitely see that they have impairment in light touch and proprioception. They lose their light touch sensation, and it's difficult for them to discriminate without their eyes open because they're losing this specific important modality of touch that is important for them to sense, and also proprioception. They are unaware of their body in space, so it's important to to have this protein in order to discriminate these two different modalities. So an important thing is that these channels are highly expressed, specifically Piezo2 in dorsal ganglion neurons. These are the primary sensory neurons. And any stimulus that comes from the periphery, it gets the information get passed on to the dorsal ganglion neurons. And from there, it gets back to the CNS where it gets processed for all the information. And it is important to point out that these are the main cells that we use endogenously to actually detect mechanical activity ion channels, specifically Piezo2. So what we know so far is that there has been many studies on Piezo channels at the biophysical and structural level in physiologically relevance, as I showed you before, but how intracellular signaling mechanisms regulate the activity, we still don't have a very clear understanding of how these channels can be regulated intrinsically. So in 2017, the lab of Arden Patapurian was able to publish a paper in which they described that activation of GQ coupled receptors can affect the activity of Piezo2 channels. So it potentiates this activity, and this potentiation is mediated by PK and PKC. Something that was not known in the literature was how these channels could also be affected by G protein coupled receptor. And we were wondering because at the DIG neuron level, we have many GI coupled receptors, and we wanted to see specifically whether GI coupled receptor can regulate the activity. So normally, GQ coupled receptor and GES coupled receptors are they mediate sensitization of neurons. However, we know by fact that GI coupled receptor, what it does is regulate neuron excitability by activating GERD channels and also regulating the activity of voltage-gated calcium channels. Something that we don't know yet specifically, or we didn't know specifically, was that activation of GI coupled receptor may also induce mechanical hypersensitivity. And this mechanical hypersensitivity was, still don't know what is the mechanism that caused that. So we wonder to see, since piezo channels were important for mechanosensation, specifically for light touch, whether activation of GI coupled receptor could affect their activity, and its affected activity, how, in what specific context. So to do that, this was the next part of the presentation, is to try to determine whether mechanically activated currents in dorsal ganglion neurons and heterologous expressed Piezo2 currents can be affected by GI coupled receptor. To do that, what we actually did was we took a mouse DRG neurons and cultured them. And after culture them, then we mechanically stimulate them. So to assess this and to see whether GI coupled receptor can regulate mechanical activity Piezo2 current endogenously, we look into the RNA level of many GI coupled receptors, and one of the highest expressing GI coupled receptors at the DIG neuron level were the GABA-B receptors. These receptors comes into fibers. They need to come into heteromerization, GABA-B1 and GABA-B2, in order to signal through GI coupled G protein. So we took this as a reference in order to study the activity of the regulated activity of Piezo2 channels. So first, what I did was that I took dorsal ganglion neurons and mechanically activate these cells with a mechanical probe. And elicit a response, as you can see here. These downward reflections are mechanically activated currents. However, when you apply baclofen, which is the activator of GABA-B receptor, over time you can see that there is a potentiation of these currents. And this potentiation was long-lasting, even more than 15 minutes. You still see this potentiation. However, it's weird. Do the same experiment, but instead of apply baclofen, which is the activator for GABA-B receptor, we apply BACO. We don't see this potentiation. So suggesting that activation of GABA-B receptor was modulating the activity of these mechanically activated currents in dorsal ganglion neurons. So if instead of using a fixed mechanically stimulus, we use an increased stimulus in which we stimulate the cell even more, so we can actually get these downward reflections. As you see, the farther the probes travels and stimulate the cell, you get more and more current coming into the cell. So I use this as a secondary method in order to test the cells. And is I apply baclofen for about 3 minutes, and then in the continuous presence of baclofen, I re-stimulate the same cell, I could still get this potentiation. And this potentiation was also long-lasting. As you can see that the effect did not watch out. So far, what I could do, it doesn't matter which specific protocol I use, I could still get this potentiation, suggesting that these mechanically activated, rapidly adapting mechanically activated currents were modulated by activation of GABA-B receptors. Something that we know of by FAT is that GABA-B receptors can be their neurons do not only have rapidly adapting currents, which have been attributed to Piezo2 channels, but they also have they also have intermediate adapting and slow adapting currents of channels of unknown identity that we're still trying to figure it out, which are the channels responsible for these currents. But in order to avoid that, what we actually did, and to determine that it was specifically to Piezo2 channels, what we did was to express this in heterologous system together with GABA-B receptor and see if we could definitely get the same effect. So I did the same experiment in which I mechanically stimulate the cell, and as you can see here, we get in hex cells transfected with Piezo2 channels, we can definitely get these mechanical activated currents. But as we activate GABA-B receptors in the same cell, we actually can get a potentiation of these currents. So the potentiation was definitely depending on activation of GABA-B receptors, and it was specifically modulating Piezo2 currents. If we did these experiments in cells that do not have GABA-B receptor but only have Piezo2 channels, we don't see that potentiation. So suggesting that activation of GABA-B receptor specifically was required for the potentiation of these currents. So another question that we had is through which mechanism of action? We know that, as I told you before, that activation of GI coupled receptor regulates the activity of GER channels by active modulating by modulating the activity of these channels, and also can inhibit voltage-gated calcium channels as well when it binds directly to these channels. We actually work mostly in this arm of the pathway because we know that adenyl cyclase is required for potentiation of Piezo2 currents in the case of GQ coupled receptor. So we wanted to focus our interest in G beta gamma mediated effects. So to actually look into how G beta gamma could potentially mediate this, and if it does, what we actually wanted to do was to use the beta adenine receptor kinase, the C-terminal domain of the beta adenine receptor kinase, which is known to block beta gamma and also block the signaling mechanisms. So to do that, what we actually did, we took this specific C-terminal domain of the which I'm gonna call it bark zinc, and transfected as well in hex cells together with GABA-B receptors and Piezo2 channels, and see whether I could block the activity of the potentiation of Piezo2 currents. As you can see here, is we record this this in the presence of GABA-B receptor, we get this potentiation. However, instead of transfecting Piezo2 and GABA-B receptor, we also transfect the beta gamma zinc, we can block this potentiation, suggesting that beta gamma was doing something that was mediating the potentiation of Piezo2 currents. So is instead of using G beta gamma, blocking the signaling of G beta gamma activating GABA B by activation of GABA B receptor, we bypass this activation and we introduce G beta gamma into the patch pipette to see whether G beta gamma was sufficient to modulate the activity of Piezo2 currents. We did these experiments in the RG neurons in which we put it into the patch pipe into the intercellular solution. We can see that there was a potentiation, that activation of GABA B receptor was not required, but we can definitely just add G beta gamma, purify G beta gamma, and we can definitely get the potentiation back. And if we have vehicle instead of that, we don't get that potentiation. Suggesting that G beta gamma was mediating the effect of Piezo2 channels currents potentiation. So to summarize this first part, what I show you is that native mechanical activated currents are, or specifically rapidly adapting mechanical activated currents, are potentiated after activation of GI copper receptor. And these piezo currents are potentiated. And something that I didn't show because of time is that while piezo1 currents are potentiated and piezo1 currents are inhibited, we believe that is also through G beta gamma, but we don't have more information about that part. And these things to the Piezo2 mediated effect seems to be mediated by G beta gamma is required and sufficient to trigger the potentiation. That we don't actually need to activate a GI copper receptor, but a G beta gamma is sufficient to trigger this potentiation. So something that I showed you before is that this was a very long-lasting effect. As you can see here, it lasts more than 15 minutes. So we thought of that normally GI-mediated effects are very quick and fast. And this is because when G beta gamma release, when activation of GI copper receptor release G beta gamma, they bind specifically to the channel. And once the agonist is not present, we can recover this. And this did not happen in the case of activation of potentiation of Piezo2 currents. So we thought that maybe the most indirect effect, and we looked to see what other mechanisms can G beta gamma affect. And we saw that G beta gamma can also activate PI3 kinase and also MAC kinase pathway. And we thought that maybe that could be an alternative mechanism of G beta gamma to modulate the activity of Piezo2 channels. In order to do that, we use blockers of PI3 kinase, we use warmanin and MAC kinase, another inhibitor of MAC kinase, to see if potentiation of Piezo2 current could be inhibited. So we treated cells either with vehicle or these inhibitors. In vehicle cells, we can see the potentiation of Piezo2 current as the baclofen application. However, in cells that were treated with warmanin, which is an inhibitor of PI3 kinase, we get an inhibition of those currents. And if we use MAC kinase inhibitor, we also get an inhibitor of this current, suggesting that MAC kinase and PI3 kinase might be involved in the potentiation of Piezo2 currents. So in order to put this into context, as I showed you before, Piezo2 currents are really important for mechanosensation. It's still a little bit debatable whether they're important for pain, but at the level of somatosensation and mechanosensation, we definitely know that piezo plays a role. So to do that, we just wanted to see if we could affect mechanosensitivity in mice by activating GI copper receptors. So to do that, in the lab, what we actually did, we used another GI copper receptor, which is called serotonin 1D. And in this receptor, we use a drug called somatriptan, which actually will activate this specific receptor. This is also a GI copper receptor. So this is the paradigm. So what we do is we acclimate the mice for about 30 minutes. And then after that, we inject somatriptan, which is the drug that activates serotonin receptors into the hippo of the animal. We also have other groups in which we also inject either vehicle, warmanin and MAC kinase inhibitors, or we put somatriptan and PI3 kinase and MAC kinase inhibitor. After 30 minutes of this injection, then we do bone-free filaments. Specifically, we're using filaments which are not going to elicit a painful response. So we use forces that definitely will only affect mechanosensitivity or only assess mechanosensitivity, but will not trigger a painful response. And then 10 minutes after, then we scored after 10 times of poking the pole, then we actually just quantify the percentage of pole withdrawals. So female mice, as you can see here, show that somatriptan treated group has an increase in percentage of withdrawal, suggesting that they're becoming more hypersensitive. However, if we actually have a group in which we have somatriptan plus mechanism inhibitors and PI3 kinase inhibitors, we can block this mechanism sensitivity, hypersensitivity, similar to the vehicle as well. And it doesn't matter at which specific force we tried, we still got the same results. That somatriptan groups will have an increase in mechanosensitivity or percentage in pulp withdrawals. It doesn't matter in females or males. Males also have the same effect. So somatriptan treated group males will also have an increase in hypersensitivity. And this increase is blocked by co-applying somatriptan together with mechanism inhibitors and PI3 inhibitors. So to conclude this part, I showed you that mechanically activated currents are potentiated after activation of G-coupled receptor, and that Piezo2 currents are potentiated. I didn't show you the Piezo1 data, but also we saw an inhibition on Piezo1 data in head cells. G beta gamma seems to be required and insufficient to trigger the potentiation of Piezo2 currents, and PI3 kinase and mechanism seems to participate in the potentiation of Piezo2 currents via G beta gamma. And something important is that activation of GI-coupled receptor leads to an increase in mechanosensitivity, and this seems to be mediated as well by PI3 kinase and mechanism. And this is actually one of the prototype models that we have that could potentially be playing a role in this scenario. So now we're going to move gear into the second part of the talk, in which we're actually trying to describe exogenous novel inhibitors of TRPV5 channels. So TRP channels in Tibor Rojas lab, they work with many TRP channels, including that they are classified in many different families. So one of the biggest families of channels that we have. We have vanilloid families. But in the lab, we basically focus on the vanilloid, the melastatins one. That's basically the ones that we actually study in the lab. And this is because these channels are important for many different physiological functions when it comes to thermosensation. We also there is TRPMA that works for actually mental. We have TRPA1, which is a chemoreceptor, and they respond to many ranges of temperature, and they can be activated by many different stimuli as well. Garlic, we can also have a chili pepper, which is TRPV1, mint. So all of these channels are important, and they are really good targets for many different physiological functions. For today's talk, we're just gonna focus on the only calcium selective, because most of those channels, most of the three channels are non-selective cation channels. However, TRPV5 and TRPV6 are calcium selective channels, and they are not thermosensitive as well. So TRPV5 channels are highly expressed in the kidney, and they are important for calcium regulation and calcium reabsorption in the kidney, while TRPV6 channels are highly expressed in the intestine, and they also regulate calcium reabsorption and homeostasis in the intestine. An important thing about TRPV5 channels is that although all the three channels are non-selective cation channels, this one is calcium selective, but also they are inward rectifiers. They require STIC2 for channel opening. They are negatively regulated by calmodulin. And an important feature that I want to point out is that they are highly homologous in protein sequence to TRPV6. And this is important because in order to find inhibitors that are specific to TRPV5 is a very challenging task because TRPV6 is extremely similar. About 70% of the protein sequence is very similar to TRPV6 and TRPV5. They shares very things in common. And as I told you, they play an essential role in calcium homeostasis, but they are also intrinsically negatively regulated by calcium as well. So we have many structures, but in 2018, we, in collaboration with Vera, Moinsin Covabel from UPenn, we got the first structure of TRPV5 in the presence of econazole. Econazole is a specific inhibitors of TRPV5, but also inhibit TRPV6. And we were able to get the structure and also do some functional experiments as well to see what was the IC50 of econazole. And we can see here that econazole had an IC50 into the micromolar range, about 2 micromolar. And as you can see here, I just gonna point out that as these channels are constantly open, they're always open, but they're regulated. They're intrinsically regulated by calcium. And once you remove magnesium, you can actually open. They're open, and as you apply econazole, you can inhibit the activity. Here I'm recording specifically lithium chloride currents, and I'm saying this because although they permeate calcium, calcium can intrinsically affect their activity, and we get very small currents. So it's better to use a lithium-based solution in order to study them. So an important thing to mention about the econazole is that econazole has a high potency in the micromolar range. However, is non-selective also for TRPV5 over TRPV6 and require high percentage of DMSO to reach solubility, which is a problem. So we wanted to see if based on this binding pocket for econazole, we could find better drug targets for this specific channel. To do that, we collaborated with a computational biophysicist, which is Dr. Marta Filisola, Mount Sinai, and her postdoc, Ajit Kapoor. So they together were able to go to a big compound library. In this compound library, they doc about 12 million compounds. From these 12 million compounds, they come out with 100 potential candidates based off of like a high score. From these candidates, there were 65 potential compounds that we could be purchased, but only 43 of them were available for purchase. So we test them all electrophysiologically. So from these 43, 41 of them showed no response. So as you can see here, some of them showed clear no effect in inhibition of this current. However, compound A, which we're going to call it today because of the large name, could inhibit TRPV5. As you can see here, this compound inhibit TRPV5, but also inhibit TRPV6. When we quantify the IC50 of this, we can see that compound A inhibit TRPV5 at about 2.91 micromolar, while TRPV6 at about 4.4, which is very close to TRPV6. So an important thing about compound A is that it requires a very low percentage of DMSO to reach solubility, but it still was its potency was in the mid-micromolar range, while it was not selective for TRPV5 over TRPV6. So what we decided to do was to see if there was a better and more efficient inhibitor of TRPV5. To do that, what we actually did was we looked into the derivative of compound A. So from this, we were able to find 5 different derivatives, and from 5 different derivatives, 4 of these derivatives did not show a clear inhibition. So, however, compound B which is the fifth compound, was able to inhibit very strongly the activity of TRPV5. And surprisingly, it did not only inhibit TRPV5, but it did not inhibit TRPV6, even to concentrations of 10 micromolar. When we calculated the IC50 specifically for TRPV5, we saw that compound B was able to inhibit TRPV5 at about 100 nanomolar. We also normally every data that I showed you before was basically using the rabbit TRPV5. However, we use to see whether we can replicate the same data in human TRPV5, and we got almost the same IC50 for human TRPV5. Suggesting that it doesn't matter between species, we still got the same potency in inhibition. So we wanted to screen for potential inhibitors, for this inhibitory effect on over 3B3 channels, and we can see that for 3B1, for 3B3, for 3B4, for 3PMA, we did not see a strong inhibition, at least in the nanomolar or lower micromolar range. However, we do see a little bit inhibitory effect 10 micromolar or in between 3B3 and 3PMA. So as I told you before, we recorded monovalent currents, basically lithium chloride currents, to actually assess the electrophysiology in 3B5 with the compounds. However, we did not use calcium in the solution, but we did actually measure calcium transients to see whether it could be inhibited, and the inhibition can only be seen specifically in 3B5 channels. So 3B6, 3B1, 3B4, 3PM3, and 3PMA did not have an inhibition of calcium transients when they were applied with the compound B, which is very specific to 3B5. So then we went in, we also and this was a collaboration between 3 labs. So in order to get the structure of this compound together with 3B5, the group of Beramo and Secova-Bell, in his also Dr. Taylor Hughes together, got the structure of this compound of this channel in the presence of this compound B. And we can see that compound B was specifically located at the S1 and S4 bundle, and also close to the trip helix. And the residues that were specifically touching this molecule, as you can see here, were D46, D406, as you can see here, tyrosine 415, and all of these residues were specifically in a different unexpected site of the 3B5, of the 3B5 channel. So what we actually did, we're trying to do some mutagenesis of these specific residues to see if we can affect the inhibitory potency. So when we mutated 415 or D46, we saw a right shift in the dose curve. And as you can see, the right shift was even stronger for D406. And we were able to shift the IC50 from 100 nanomolar to 3.68 micromolar in D406, and for Y406 about 600 nanomolar. So suggesting that this could be potentially the binding pocket for compound B. So to conclude that, we can actually say that we reported in the paper, we reported 3 novel 3B5 inhibitors. I didn't show data here because of sake of time, but we published 3 novel three novel TRPV5 inhibitors. The other inhibitors, we didn't characterize it further because it was very similar to compound A, which was non-specific, and it seems to have a very high it seems to have a very high a concentration in order to reach an inhibitory effect. So it was very similar to compound A. So for compound B, we saw that it was very specific to TRPV5 over TRPV6, and structural and functional characterizations of compound B identified that the site was binding into the S1, S4 bundle in the trip helix of TRPV5. And this later binding site is partially overlapped with modulators of TRPMA and TRPV6 structure as well. We believe that the novel inhibitor is locking TRPV5 in a very close state. So once it's binding to maybe clouding the pore, no allowing ions pass through. With that, I want to acknowledge a lot of people that worked through this. So I have many collaborators from my first project, Piezo2, and also for the TRPV5s, Dr. Marta Felizola, Vera Monse-Cobabel, Dr. Taylor Hughes, and Dr. Ajit Kapoor, and also the people that works with the Piezo2 channels, which is actually, I wanted to thank Dr. Taud and Dr. Micharhi and Cassandra Hartle as well for helping me out with this part. Of course, the lab member in my postdoc, my PhD mentor, Dr. Tibor Rohach, and my lab members at Rohach Lab as well. And of course, the grants from NIH that has been able to support me throughout my work as well. So now I'm willing to take some questions if anybody have questions. Thank you so much.
Thank you very much for an excellent talk. The panel is now open. Actually, I got a quick note regarding the inability for one attendee to post a question. So if there's anyone who is wanting to post a question and has a problem, please push the raise hand button, and we'll try to either unmute you or be able to somehow get that question. But nonetheless, we do actually have questions, and I'm going to start off one with my own, and which is, can you speak to Dr. Rosario? Can you speak to the expression pattern in human DRG?
Of Piezo2 channels?
For actually of both channels, but let's start with Piezo2.
So there hasn't been a lot of studies about the expression of Piezo2 channels. I cannot comment a lot onto that. This is an area that definitely needs to be further researched. I do know that Alex Chesler from the NIH, he has been able to get to transform stem cells into IPC cells into DRG neuron-like from donors that have mutations on Piezo2. But so far, that's basically what has been done into detect Piezo2 currents in human tissue, but that I know of like expression patterns of Piezo2 in human DRGs, it has not been done. And I think this is the great time to do it, specifically that now AnaBios has the possibility of providing human DRGs to do those things. For TRPV5, I know there are many mutations, but how highly expressed? I do know that they are highly expressed in the kidney, and they regulate kidney homeostasis, and mutations in the channels can affect the concentration of calcium in the kidney. But basically, TRPV5 is not expressed in dorsal root ganglion neurons. It's only expressed in these specific non-excitable cells. located in the kidney, and also in the, you can also see it in the intestine, but preferentially 3B6 is there. So that's basically what I think the literature has pointed out so far.
Thank you. I have some questions from the audience. So the first question, which also has a second question on TRPV5, but we'll start with the terms of piezo. So are GABA A receptors and piezo channels co-expressed in DRG neurons? And if so, how does GABA-dependent depolarization of the DRG neurons affect piezo currents and their GI modulation? This questioner points out that the GABA in this context is a depolarizing neurotransmitter.
Okay, so I would say it like this. This is gonna be a pretty educated guess answer that I'm gonna give, because specifically GABA A receptor are ionotropic receptors compared to GABA B receptor, which are metabotropic receptors. So GABA A receptor, the expression, I do believe they are expressed in the DRG neurons. I can comment on what the depolarization of the cell by GABA A receptor activation will lead to Piezo2 channels activity. I do know that Tibor Rojas previously published a paper in which he's described the activation of 3B1 channels, which are non-selective cation channels, can inhibit the activity of Piezo2 channels. So I will say that an incoming increase in the polarization of the cell could potentially inhibit Piezo2 channels. It's been shown at the level of the ear, that in hair cells, activation of calcium can inhibit 3-Piezo2. So I would guess that activation of this will actually affect the activity of channels, but not in the potentiated part, but by modulating their activity, by inhibiting the activity of these channels. So that's basically what I can comment when it comes to the GABA A receptors, because they are completely different. GABA A receptor will not lead to a signaling cascade of beta gamma activation, but it will just depolarize the cell, and that definitely resembles to what happens with 3B1. That could potentially resembles that.
Right. I'm going to ask the follow-up question from the listener, which is regarding your TRPV5 data, which is, why does the compound B induce calcium transients on their own?
No, maybe I didn't. It was a little confusing. Calcium B do not produce calcium transient. I was just this cell has been transfected with a GCAM, and what we're measuring is once with so these channels are blocked by magnesium. So when we remove magnesium from the solution, they are constantly open. They will open, and in the solution we also have calcium. So calcium will come in, and then you will see calcium transients that are going to be read out based on the GCAM signal, and this signal can be blocked by compound B because it will close the channel. So it's not like that calcium. It's not like compound B will block, will cause calcium transient, but it will block the activity of the channel, and then by blocking it, it also affects the calcium coming into the cell and producing the calcium transient that we see. Does it make sense?
I think you may have to follow up with the listener. The listener does point out that the calcium signal increases as soon as you apply compound B. So what we might do, if you want to respond to that, and then maybe we can have a conversation afterwards.
Okay.
So what I'll, the next question is a great talk. In your human DRG work, did you characterize different mechanical currents subtypes? In other words, did you see slowly and intermediately adapting mechanical currents like those similar seen as you observed in the rodent DRG?
So most of the work that I have here is not in human DRG. The work is entirely in mouse DRG. And I could not, in the mouse DRG, I basically saw rapidly adapting mechanically activated currents. And this is because I was specifically selecting large dorsal root ganglion neurons, which are mechanoreceptors. So I was targeting the population of cells that basically was activating rapidly adapting mechanical activated currents. So the majority of the cells that I saw, I would say 95% were rapidly adapting mechanically activated currents.
Thank you. As a follow-up question, actually, on the piezo and the GABA-B, I was going to note that we've done some, AnaBios did some work with Stuart Brary showing that baclofen actually is reduces neuroexcitability. So I was interested in terms of what you thought of the alignment of the GABA-B and the Piezo2. Are they perfectly aligned, or do you have situations where GABA-B is in the case of the mouse, of course, are GABA-B and they're not in the same neurons, and they could have differential effects?
Something that I didn't mention is that definitely it could be the case that some neurons might not have GABA-B. So GABA-B has to come into different, it has to come into heteromer in order to produce the signaling. And not every cell that I was able to apply GABA-B, I saw the potentiation. I didn't show this just for the sake of time, but yes, some cells do not have when I apply. They do have Piezo2 channels, but they might not have. I didn't see the potentiation, and I attributed this specifically to not being able to express both channels in the same cell. When it comes to like whether, can you just repeat the second part you said that? Can you just repeat?
I'm sorry.
You just mentioned that is this changes the depolarization? What were you saying?
Oh, right. So what we observed is that the GABA-B does depress neuroexcitability. And since in your data, it seems to potentiate the Piezo2 current, which I presume is an excitatory role in these neurons, so that there's potentially, and I think you're alluding to this already, is that there's not perhaps a perfect alignment. It depends upon whether or not they're co-expressed or not, or are not co-expressed.
Exactly. So it depends basically that they are not completely co-expressed together. And I didn't mention these for the sake of time too, but I also tried serotonin receptors in a specific serotonin receptor D, and I also saw the potentiation in about more than 50% of the cells. So suggesting that not every cell will have the same characteristics. We're talking about human mouse DRG, and these mouse DRGs are very heterogeneous. So it's difficult to assess what is the excitability property of each cell, because we don't know what is the composition of each cell. And since I didn't have a specific marker to target with these specific cells, we have this excitation profile, or this one will have this inhibitory profile. So basically, I just wanted to use baclofen as a reference point and to actually study the signaling, which is highly expressed in many cells that I was looking at.
Thank you. Understood. Do we have any other questions? Well, John, thank you very much. It was an excellent talk, and I want to also thank all the listeners and audience members for having joined us today. And we appreciate your joining all our webinar series. So if you'd like to also download and listen again to the talk, this talk is being recorded and will be made available at the AnaBios website. So for now, I'd like to thank you again, and I hope to see you, or I hope you'll be able to join us in one of our next webinars. Thank you very much.
Thank you so much. Thank you.